Introduction. What is the cell cycle? The cell cycle is a series of events that occurs in a cell leading to its growth, DNA replication, and division into two daughter cells. It is a highly regulated process that ensures the accurate transmission of genetic information from one generation of cell to the next. The cell cycle plays an essential role in growth, development, tissue repair, and the repayment of damaged or dead cells. Proper regulation of the cell cycle is critical for maintaining normal cellular function, preventing disease such as cancer. Main function of the cell cycle: growth and development, repayment of all damaged cells, maintenance of genetic stability, tissue repair, and regeneration.
Interphase, the preparation phase where the cell growth and replication is DNA. Key feature: longest phase of the cell cycle, about 90%, cell growth and normal metabolism, protein and organelle synthesis, DNA replication, and preparation for mitosis. Three-stage interphase: G1 phase, cell growth; S phase, DNA replication; G2 phase, preparation for mitosis.
Interface G1 S G2 G1 the cell growth and synthesis RNA, protein, and enzyme needs to prepare for DNA replication. S the cell replicates its DNA, doubling the DNA content, while synthesis histone and duplicates centriole in animal cell. G2 the cell continues to grow, synthesis addition the protein, and prepare for entrance into M-phase or mitosis.
M-phase, mitotic phase or M-phase. The mitotic phase includes two overlapping processes: mitosis, in which the nucleus and its contents divide evenly into two daughter nuclei, divided into 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. Cytokinesis, in which the cytoplasm is divided into daughter cells, occurs during telophase. Cytokinesis in animal cells. In animal cells, cytokinesis occurs by a process known as cleavage, forming a cleavage furrow. Cleavage furrow is formed by the contraction of contractile ring. The cleavage furrow deepens until the parent cell is pinched in two, producing two completely separate cells. Cytokinesis in plant cells. In plant cells, a cell plate forms during cytokinesis. Vesicles containing cell wall materials derived from the Golgi apparatus move along microtubules to the middle of the cell, where they fuse, producing a cell plate.
The next one is mitosis. First, prophase. Chromatin fiber condensed into discrete chromosomes. Nucleolus disappears. In animal cell, microtubule grow out from each centrosome, forming ester and mitotic spindle. Centers from each other to opposite ends of the cells. Second, metaphase. The centrosomes are now at opposite poles of the cell. The chromosomes have become completely condensed. All chromosomes have arrived at the metaphase phase. All kinetochore to kinetochore microtubule coming from opposite poles. Third, anaphase. coenzyme is removed, turning sister chromatids into independent chromosomes. They move centromere first to opposite pole as kinetochore microtubule shorten. The cell elongate as non-kinetochore microtubules lengthen. The last one, telophase. Nuclear envelope arise. Two daughter nuclei form in the cell. Nucleoli reappear. Chromosomes begin to relax. Any remaining spindle fibers are depolarized.
Cell cycle checkpoint. What are cell cycle checkpoint? Cell cycle checkpoint are control mechanisms that assure a cell is ready before moving to the next stage of the cell cycle. They help maintain DNA accuracy and prevent damaged cells from dividing. Three main checkpoints. 1. G1 checkpoint. Check nutrient and cell size and detect DNA damage before DNA replication. 2. H checkpoint. Monitor DNA replication and stop replication if errors are detected. 3. T2 checkpoint. Confirm DNA replication is complete and check DNA integrity before meiosis.
Next, I will talk about the G0 phase. The G0 or gap 0 phase is a resting state where cells leave the active cycle. During this phase, cells stop preparing for division, but they remain metabolically active and continue performing their normal functions. There are two types of G0 phase. The first is temporary or reversible G0. These cells can return to the cell cycle when they receive the right signals, such as tissue repair. Examples include liver cells and lymphocytes. The second is permanent or irreversible G0. These cells permanently lose the ability to divide. If they are damaged, they are difficult or impossible to replace. Example includes neurons and cardiac muscle cells. G0 phase is important because it prevents unnecessary cells division and helps maintain normal tissue function and cellular specialization. To conclude, the cell cycle is a highly regulated process that controls cell growth, DNA replication, and cell division. During interphase, the cell grows, copies its DNA, and prepares for division. Then, in the M phase, one parent cell divides to produce two daughter cells. Throughout the cell cycle, checkpoints ensure that each stage is complete equally before the cell moves on to the next step. Some cells may also enter the G0 phase where they stop dividing but continue carrying out their normal functions. Overall, proper regulation of the cell cycle is essential for normal growth, tissue maintenance, and preventing abnormal cell proliferation, which can lead to diseases such as cancer. Thank you for your attention and listening.